This video is all about HIV. So this is for you AQA biologists. Now I'm filming this now because we do viral structure quite early on in year 12, but you might not actually learn about HIV specifically or how it replicates until you do immunity later on in the year. But obviously if you're year 13, you can use this for revision because HIV replication is one of the topics that students really struggle with. Let's introduce HIV. So HIV stands for Human Immunodeficiency Virus. That's where HIV comes from. It causes AIDS, which is the disease caused by the virus. Now, AIDS stands for autoimmune disease because this disease basically is destroying and weakening your immune system. Specifically, HIV destroys your helper T cells, which are a type of lymphocytes involved in your specific immune response. Now, if HIV infection is present and HIV is destroying helper T cells, what would be the effect on the immune response? So let's kind of revise immunity a little bit here. So if we're destroying helper T cells, we're going to have fewer helper T cells. And that means we have fewer helper T cells to activate the B cells or the B lymphocytes. So we can say fewer B cells or B lymphocytes will be activated or stimulated. Because remember, one of the jobs of the helper T cell, as well as to stimulate phagocytosis, is to stimulate or activate the B cells to divide by mitosis. So if fewer B cells are activated or stimulated, then fewer B cells divide by mitosis, or we can call it clonal expansion. Okay, because there's fewer B cells to activate them. Sorry, fewer T cells, fewer helper T cells to activate the B cells. If fewer B cells are being stimulated or activated, they are not going to clone themselves or divide by mitosis or undergo clonal expansion. So then we're going to have fewer plasma cells. Because once we activate the B cell using the helper T cell and it divides by mitosis, these cells then differentiate, don't they, into plasma cells or memory B cells. Now, if we end up with fewer plasma cells, we're going to have fewer antibodies produced, which is kind of the end place we need to get to, right? Fewer antibodies produced. So we have a weaker immune system or a reduced immune response. Now, this is kind of good revision, right, on immunity if you have done this already. With helper T cells being destroyed, fewer helper T cells to activate the B cells. So fewer B cells divide by mitosis or undergo clonal expansion. Fewer B cells will differentiate into plasma cells. Fewer plasma cells to make antibodies means less antibodies. And basically, we've got a far weakened immune response, okay? And AIDS occurs when the helper T cell number has got low enough that this individual is then going to suffer from lots of infections that could potentially be fatal because the immune response is so weak. Let's have a look at the structure. Now, we did viral structure in a previous video that we posted recently, but that was kind of general viral structure. Now we're thinking specifically about HIV as a virus. So it has attachment proteins on the surface, and they are there to attach to receptors. Now, in general, we could just say attach to receptors on host cells, but we know that HIV infects T helper cells. So I'm going to say to attach to receptors on helper T cells. Okay, they're actually called CD4 receptors. You don't have to know that, but you might do. So I'll write it in. So yeah, the attachment proteins on the surface of the HIV virus attach to CD4 receptors on helper T cells. The capsid, which is made of protein, protects or stores the viral, I'm going to put RNA because this is HIV, right? And HIV has RNA as its genetic material, not DNA. So it'll be RNA that's in the capsid if it's specifically HIV. So the nucleic acid, which we've just said is RNA, codes for viral proteins, or again, we could be specific and say HIV proteins. And HIV does need reverse transcriptase, which we can see here, these little pink enzymes. And the reason it needs these enzymes is because these enzymes convert the RNA into DNA. Because ultimately what the virus wants to do is get its genetic material into the host cell's DNA. Now, because the host cells have DNA, HIV can't insert RNA into DNA. So it uses reverse transcriptase, the enzyme, to convert its RNA into DNA so it can be incorporated into the host cell's DNA. Now, not all viruses have reverse transcriptase because not all viruses have RNA. Some have DNA and therefore they don't need reverse transcriptase. HIV infection. So as we've already said, HIV infects a particular type of immune system cell. It's the T helper cell. It's the CD4 plus T helper cell, but at A level, we can just say it infects T helper cells, which are one of the lymphocytes, the T cells, the lymphocytes involved in a specific immune response. Once the person is infected with HIV, they are said to be HIV positive. And because the T helper cells are infected with HIV, they are going to turn into HIV replicating cells. So the HIV has basically hijacked these T helper cells and is using them to make viral proteins, which are then going to be assembled into new HIV viruses, which are going to burst out of the T helper cell. And in doing so, they're going to destroy it. There are typically 1 million T helper cells per 1 milliliter of blood. Don't need to know that. But what HIV does is it will slowly reduce the number of these T helper cells. And once that number gets low enough, we say that the person has developed AIDS because their immune system is considerably weakened and they are at risk from other infections. How does HIV replicate? Now, this is a simple overview, but on the next page or the next slide, I'm going to give you a mark scheme answer. So do continue watching. If we start here, we can see that the HIV virus has used its attachment proteins to attach to the receptors on the lymphocyte. And remember, this lymphocyte is a T helper cell or a helper T cell, doesn't matter which way you say it. And the attachment proteins are attaching to the CD4 receptors on the surface of that T helper cell. The HIV then injects its genetic material into the lymphocyte. So it's becoming a host cell, as we describe it. Remember, reverse transcriptase does need to convert the viral RNA into DNA before it can actually be inserted into the host cell's DNA. The HIV is then using the lymphocyte's cellular machinery to make copies of itself. So it's using the cell to make viral proteins, which will then be assembled into new HIV viruses. And after making many HIV viruses, they're going to burst out of that T helper cell and destroy that T helper cell. And obviously they're then going to go on to attach to and infect more T helper cells. Now, how are we going to write this in an exam? Could be worth 4 marks. How does HIV replicate? Let's look at a mark scheme. So we can get a mark for saying attachment proteins on the HIV attach to receptors on helper T cells. I'm going to put this in brackets because you don't need to remember it, but you do need to know it's attachment proteins on the HIV attaching to receptors on helper T cells. Once that attachment has taken place, we can say the virus fuses with the cell membrane and the viral RNA is going to enter the T helper cell. They'll even allow you to say that it injects the viral RNA into the T helper cell. Don't forget, reverse transcriptase, that enzyme is needed to convert RNA to DNA. So that's a nice easy mark that you can get. And once we've got DNA, it's incorporated or inserted into the helper T cell DNA or into the helper T cell's nucleus. Now that uses an enzyme called integrase, but I'm going to put that into brackets because you don't actually need to know that. The DNA is then transcribed. And the most important thing to say is viral proteins will be produced. And then once we've produced all the viral proteins needed to make a new HIV virus, viruses are assembled and released from the helper T cell which causes the helper T cell to burst. Obviously, those viruses can then go on and attach and infect other helper T cells. The process repeats until the number of helper T cells gets so low that we say the person has AIDS. Okay, now the 6 kind of marking points here. This is usually only a 3 to 4 mark question. So I would focus on the attachment proteins attaching to receptors on the T helper cell, the reverse transcriptase converting RNA into DNA, the DNA being inserted into the T helper cell DNA, and then viral proteins being produced and viruses being assembled. That's the thing we see repeated on lots of mark schemes over previous years. I hope you found this video useful. Stay tuned because we try and post at least 2, maybe even 3 videos every week to help you with your A-level biology.